Morning, everyone, and thank you for being here. My name is Cristina Martinez. I'm a research associate in the Clinical Ophthalmology Research Group at the University of Oxford. I'd like to thank the Macular Society for inviting me today and to thank all of you for taking the time to join. The work I will share with you today is not only about understanding a gene, it's about translating that knowledge into treatments that can preserve vision and quality of life for people living with RPGR-related conditions. Over the years, meeting people affected by these conditions has shown me how important it is to protect the vision that remains, not just to slow the disease, but to maintain independence in everyday life. That's the goal driving this research. Today, I will take you on that journey, starting with how the healthy retina works, what happens in RPGR-associated disease, how we investigate it in the lab, and what it means for future treatments like gene therapy. But first, a little bit about myself. I grew up in Valencia, a beautiful city on the eastern coast of Spain, and studied biotechnology at the university there. During that time, I started working in a hospital research lab and was introduced to retinal diseases like retinitis pigmentosa, which can lead to blindness. Meeting patients and families affected by these conditions inspired me to dedicate my career to finding new treatments. I went on to complete a PhD in biotechnology in 2015, studying what causes retinal cells to become damaged, trying to answer the question, why people go blind? Then the question changed to, what can we do to stop people going blind? And in 2016, I moved to Oxford to join Professor Robert McLaren's team, where I've been working ever since. My research focuses on developing gene therapies. This is treatments that use harmless viruses to deliver healthy genes into the retina. Some of this work has already led to a new gene therapy for X-linked retinitis pigmentosa, which is now being tested in phase 3 clinical trial and showing very encouraging results. To understand RPGR-related disease, we first need to know a little bit about the retina. So the retina is the thin, light-sensitive layer at the back of the eye. You can think of it like the film or the sensor in a camera. It captures light and turns it into signals that the brain uses to create vision. Inside the retina are two main types of photoreceptor cells, rods. These are found mostly in the peripheral retina. They allow us to see in dim or dark places. They are what give us our night vision and help us detect movement in our side peripheral vision. And then we have cones. These are concentrated in the center of the retina, especially in a region called the macula. Cones give us sharp vision in daylight and allow us to see colors. At the very center of the macula is the fovea, which is packed with cones and give us our most detailed vision, like when we are reading, recognizing our face, or seeing small details clearly. Together, rods and cones work as a team. The rods help us adapt to low light, while the cones give us color and detail in bright light. This combination allows us to see clearly in color and across different lighting conditions. But what happens when these photoreceptor cells stop working? Well, that's where genes like RPGR come in, and that's what we will explore next. So here's the journey from DNA to vision. Every cell in our body has an instruction manual. That's our DNA. This manual is divided into chapters called genes. Each gene gives the recipe for making a specific protein, and those proteins do most of the work that keeps your cells alive and healthy. Just like in a cookbook, if a recipe has a typo, the dish might turn out wrong. If a gene has a change, what we call mutation, the protein it makes might be missing, faulty, or made in the wrong amount. That can cause the cell to struggle or stop working properly. In the retina, that is a light-sensitive tissue in the eye, there are thousands of different proteins that help our photoreceptors, our rods and cones, capture light, process it, and then send the signal to the brain. If even one of the genes for those proteins doesn't work as it should, the affected cells can become sick and eventually die. In inherited retinal diseases, the problem starts with a specific gene that isn't working correctly from birth. Different genes control different steps in the retina's functions. So depending on which gene is affected, you can get different types of retinal disease with different symptoms, age of onset, and speeds of progression. Each gene plays a different role in the retina, and the type of mutation can lead to different symptoms and disease courses. These diseases are rare, but have a big impact, causing progressive vision loss and sometimes blindness. There are many different forms. We have retinitis pigmentosa, control dystrophy, macular degeneration, and others, and many different genes can be involved. Today, we'll focus on one of them, but it's RPGR. RPGR-associated retinal dystrophy is one of the most severe inherited retinal diseases. It is caused by changes, by mutations in the RPGR gene, which is located on the X chromosome. Mutations in RPGR are responsible for over 70% of X-linked retinitis pigmentosa cases and up to 20% of all retinitis pigmentosa cases overall. RPGR-related disease can vary a lot. In some people, it affects mostly the rods, causing night vision and peripheral vision problems. In others, it affects mostly the cones, leading to difficulty seeing colors and fine details. Some patients experience a combination of rod and cone problems, which can affect both night vision and sharp central vision. Retinitis pigmentosa is one of the forms that is caused by RPGR mutations. It's a group of diseases where photoreceptors gradually degenerate. It's one of the leading causes of inherited blindness, affecting about 1 in 4,000 people worldwide. Typically, RP starts with night blindness, as rods are affected first, and later progresses to affect cones. So rod cells are affected first. There is like a progressive loss of peripheral vision, leading to what is known as tunnel vision. And then cone cells also degenerate, leading to loss of central vision. And at the final stage, many patients retain some light perception for years, but some become legally blind. We have then the other form that is caused by RPGR mutations, that is cone dystrophy, where cone cells in the retina gradually lose their ability to work. And what happens is in cone dystrophy is that first central vision fades. The center of what you see can become blurry or having missing spots. Colors lose brightness, reds, greens, and blues can look faded or wrong. And bright light becomes uncomfortable. Many people find they need to use sunglasses even indoors on a bright day. Over time, some people also develop night vision problems if rod cells become involved as well. So it's usually caused by genes, by changes in mutations in specific genes, like for example RPGR, that give instructions for keep cone cells healthy. These changes mean the cone cells can't function properly, and over year they slowly wear out and die. So why it matters to study it, so understanding which gene is affected and how helps researchers develop better treatments and design gene therapy trials, for example. So cone dystrophy is quite rare. Only a few people out of every 30, 40,000 have it. In those with RPGR changes, most have a rod-first disease called retinitis pigmentosa, as we have seen before, but a smaller group, like 10, 20%, have a cone-first disease like cone dystrophy. RPGR is a gene, as I said, found on the X chromosome. This is one of the 2 sex chromosomes we inherit from our parents. Males have 1 X and 1 Y chromosome. Females have 2 X chromosomes. Because males have only one X chromosome, if their RPGR gene has a mutation, they don't have a second copy to make up for it. Females have two X chromosomes, so they often have a working copy that can help protect their vision, though some may still have symptoms. Most RPGR mutations happen in a special part of the gene called OR15. This is like a fragile chapter in the instruction manual, very long, repetitive, and prone to typos. Because OR15 is tricky for cells and for scientists as well to read, it's more likely to have changes that cause disease. Mutations here often lead to either the rod first or cone first forms of RPGR disease, depending on exactly where in OR15 the change is. One of the big challenges in studying RPGR and in designing treatments for this disease is that OR15's unusual structure makes it harder to analyze and repair. RPGR is crucial for photoreceptor cells. In the retina, rods and cones have two main parts, one that catches light and the other one that processes it. They are linked by a tiny bridge called the connecting cilium. The RPGR protein works right at that bridge, directing traffic so important materials can move where they are needed. If RPGR is damaged, that traffic system breaks down, and over time the photoreceptors cannot survive. Interestingly, some mutations affect rods more, others cones more, and this difference is important for predicting how the disease will progress. The RPGR protein has a special tail with repeated building blocks that can carry, let's say, like chemical decorations, and this is called glutamylation. These decorations help the protein control traffic inside the photoreceptor's bridge. Mutations in OR15 can disrupt these decorations, meaning the protein cannot do its job properly, and over time, the photoreceptors become unhealthy. Different changes or mutations in the RPGR gene can lead to different forms of disease. We call this genotype-phenotype correlation. So when changes happen near the start of OR15, this is more often linked to rod first disease. When changes are near the end of OR15, so these are more linked to cone first disease. And knowing exactly where the change is can help us predict symptoms, progression, and the best way to monitor the disease. Most people with RPGR mutations first notice night blindness and peripheral vision loss, but a small group has a cone first form of the disease, affecting cells, again, we use for detail and color vision. This type is less common, so we know less about how it develops and progresses. Without this knowledge, it's harder to design effective treatments and to know the right time to intervene. So what we want to find out is when the cone cells start to show damage, what early signs can we detect before too much vision is lost? How do different mutations in RPGR, especially within OR15, lead to cone first disease? We investigate this disease by eye imaging and also genetic testing, so reading the DNA recipe to find typos. We identify the exact RPGR mutation and link its location, for example, if it's at the start of the or the end of the OR15 region, to how disease appears in patients. In the lab, we use cell and animal models to help us understand what goes wrong inside photoreceptors and also to test potential therapies before human trials. Ultimately, this research gives future therapies the best chance of success and ensures patients get them at the right time. My research focuses on the form of RPGR disease where the cones are mainly affected. This is especially important because cones are critical for daily activities like reading declines in faces and again simplifying details. In our studies, we looked at several different RPGR gene variants or mutations and ask, how do these changes affect the RNA and the protein that the gene normally produces? Do these mutations completely stop RPGR from being made? Or is there still some leftover function that we might be able to use or support with future therapies? By answering these questions, we can better understand how the disease develops and how to design treatments that target what is left of the gene's function. To study how RPGR mutations affect patients, we need to look at how their cells make RNA and protein. Of course, we cannot easily take samples from the retina, so instead we use a much less invasive method. So we take a tiny skin sample from the patient. From these, we grow cells called fibroblasts in the lab. These cells carry the patient's own genes, so they act as a window into how those genes work. And then using these patient-derived cells, along with other laboratory models, we then extract RNA. This is the instructions that carry the gene's message. We extract the protein, which is the final product made from those instructions. We measure how much RPGR, RNA, and protein is produced. By comparing the RNA and protein levels, we can see whether a genetic change blocks the process at the RNA stage. This is the message is missing or faulty. At the protein stage, the message is there, but the product isn't made, or at both stages. And this step-by-step approach helps us understand exactly where the problem lies, which is essential for designing the right kind of therapy. And here, just to show you an example, so we studied two brothers with RPGR mutations who developed night blindness in early adulthood, but surprisingly, their central vision, the vision we use for reading and color, remained excellent. This is very unusual for RPGR disease at their age. When we look at their eyes with imaging, we saw advanced damage in the peripheral retina, but the central part of the retina, the macula, was largely spared. Functional testing showed that while there were some subtle losses, the central vision points were normal in both brothers, even though the gene mutation was expected to harm cone cells. This shows that cone function can be remarkably preserved even when other parts of the retina are severely affected. Genetic testing showed that these two brothers have a change in their RPGR gene that affects the main versions of the protein. And this change is like a small typo in the genetic instructions. It removes 2 letters from the code, which causes the protein to be cut short. Normally, the missing part includes a region that is very important for cone cells, so we will expect the cells to be damaged. But in these brothers, the cone cells are working surprisingly well. This made us wonder, like, how are the cone cells still functioning? Is there like a backup mechanism in the gene, like an alternative way of reading the instructions that partially rescues the protein's function? And to understand why these brothers' cone cells are working so well, we use computer tools to study the gene. The analysis suggested that the small genetic change might create a kind of backup plan in the instructions, a way for the cell to skip over part of the code while still keeping the important end portion intact. To test this, we look at the gene activity and cell taking from one of the brothers. We found 2 types of messages being made, one with the original small deletion and one that looks like it followed the backup plan. And then we compare with healthy control cells that only have the normal message. This means that even though the gene is damaged, the cells may be able to produce a partly functional protein. This could explain why cone cells are preserved despite a mutation that we expected to be harmful. Even when the RPGR gene is damaged, the body can sometimes use clever backup mechanisms to keep cone cells working. And studying these rare cases helps us understand the disease better and guide future treatments to protect or restore vision. Right now, there are no cures for RPGR-associated retinal dystrophy, but gene therapy offers real hope. The idea is simple. If a gene isn't working properly, we try to give the cells a healthy copy so they can function again. You can think of it like an instruction manual again. If the original page has a mistake, a mutation, the cells cannot follow the instructions correctly. With gene therapy, we deliver a clean copy of the page so the cells know what to do again. And here's how it works. We take a healthy copy of the RPGR gene, in this case, we package it inside a harmless delivery vehicle called adeno-associated virus, which has been specially modified to carry the gene safely. This vehicle is then delivered into the retinal cells, where the new gene provides the right instructions to make the missing or faulty protein. The goal of gene therapy is to keep the photoreceptor cells alive and working for longer, slow down or even stop the progression of vision loss. One very important point here is that early diagnosis makes a big difference. The therapy works best while the cells are still present and can be rescued. There are now 2 advanced clinical trials testing gene therapy for RPGR-associated retinal disease. Both aim to protect photoreceptor cells, but they use slightly different approaches to deliver the gene. Think of it like sending a replacement instruction manual to the cells. Right? So one therapy uses a slightly shorter version of the RPGR gene. It's missing a small piece, while the other uses the full-length gene that has been optimized so the cell can read it efficiently. Even small differences in the gene can change how well the therapy works. And because these treatments are given only once directly into the eye, it's really important to understand exactly what each therapy delivers. So while both approaches are promising and aim for the same goal, which is preserving vision, they are not identical. And scientists are carefully studying how these differences affect safety and effectiveness, which help us design better treatments and give patients the best chance of preserving their vision. So to bring it all together, RPGR-associated retinal diseases are rare but serious, and they affect people in different ways, depending on which cells are involved. Studying these diseases carefully, from the genes to the cells to vision tests, help us understand how and when the retina is damaged. Every mutation in RPGR can affect the retina differently. So each patient situation needs careful study. There isn't a one-size-fits-all approach. Understanding the specifics of each case helps us design the most effective treatment. This knowledge guides the design of gene therapies, allowing us to target the right patients at the right time and to create safer and more effective treatments. Even in cases where the gene is damaged, the body can sometimes compensate, giving important clues about how to protect vision. By combining clinical studies, genetic analysis, and lab research, we are building the foundation for treatments that could preserve or restore sight for patients with RPGR disease. We are not just studying mutations, we are building the knowledge that will lead to better and safer and more effective treatments. I would like to thank my colleagues at the University of Oxford, our collaborators, and our funding bodies. Most importantly, I want to thank the patients and families. This research is done to help fight this disease and to work towards better treatments and a better future for your vision. And thank you all for listening. I'll be happy to take questions.
Cristina, that was fantastic. Thanks for joining us today. Now, unfortunately, you were unable to join us live, so we've got a few questions for you that we think that some of our members and people watching might want to ask. So we'll just fire away. So the first question: do we know why RPRG, IPR, IP, RPGR mutations can lead to such various different conditions? So do we know why there's such a spread of conditions through the damage to this gene?
That's a good question, and that's what we are investigating. So by studying like over 100 patients, we have seen that there is like a clear, let's say, distribution. So when changes affect one part of the gene, they are more likely to affect rods. And when changes affect the other part of the gene, are more likely to affect cones. And we are studying why that is happening. And we believe that one of the reasons is that the end of the gene that is affected and is causing like cone dystrophy, it's basically all these chemical decorations, all these tags I was talking about, that's impaired. So when the gene like don't have like these tags, that affects cones more than rods. So we are exploring the molecular pathways, like the reason like why these tags are so important for cones. But we know there's like a clear, as I mentioned in the talk, like a gene type phenotype correlation. Like depending on where the change is, it affects one type of cell or the other. And we are still working on that, but we believe that those chemical decorations, like the glutamylation, is what is really important for cone function.
That's great. I think it leads beautifully on to our next question. Funny enough, so is genetic testing a way of predicting the type of condition, and then potentially how the condition will progress?
Testing is critical. We need to know where the change happens. And with the genetic testing, we get the answer like this is the mutation, this is the gene, this is the typo in the recipe that is causing the problem. And from there, we study, okay, so it's in this position, it's affecting this part of the gene, it's affecting this other part. We usually do like different computer analysis to try to predict the impact of that typo on how the dish turns out, like how the protein is made. So genetic testing is essential. We need to know what is the change. And then from there, we can try to predict what will be the impact of that change in rods or cones and what can be the progression of the disease. Yeah.
It's a question we get asked an awful lot at the helpline and across the society about genetic testing. So it's always good to hear that it's quite essential. Even though it does take quite a long time to get the responses back, doesn't it? But it's worth it in the long run. So that leads sort of onto the another question around gene therapies. Then, so you mentioned gene therapies in your session. So realistically, are we getting closer to treatments and for patients that can see benefit to patients?
Hopefully, yes. So there are currently several gene therapy studies. Two of them have just completed phase 3, which is the most advanced phase of the clinical trial. So one of them unfortunately didn't meet the primary endpoint, so didn't get the results that were kind of expected. And I understand they are in the process to see if this could get into the market at some point. Then we have this other trial that is also into phase 3. So this company completed enrollment like a couple of months ago, and they hope to have the results ready by next summer, so 2026. And the results look promising, look encouraging, but we need to wait to have the results and to see if we can meet that threshold in order for the therapy to move forward and hopefully get approved in a few years. Let's hope so. And then there are other trials as well that are like in phase 1, phase 2. They are not that advanced, but they are also exploring gene therapy vectors for RPGR. But yeah,
that does sound very encouraging. We always ask this question. We're always ask this question through the helpline. Gene therapy does feel like it's going to be the way forward. Which is very exciting for people living with all sorts of retinal conditions. So that, which actually, so you mentioned in your presentation actually using someone's cells to sort of have a look for the damage to the genes or the mutations. So how can people get involved in the sort of genetic test, not genetic testing, research and trials with cone dystrophies if they have a cone dystrophy?
So, I mean, patients can contact, like for example, in our group, we work in the Oxford University hospitals, in the eye hospital. So they can always contact like Macular Society, or they can contact our department, just checking like the website and the inquiries email address. And we have several studies where we really need participation of patients to be able to understand how these changes affect the cells in the patient's body. So this is something that can be checked online in our website or with Macular Society.
Brilliant. Okay, well, fantastic. We do have our Macular Society database and our fantastic research team that will always be happy to answer some of these questions as well. So thank you for that. So my final question. So so if people want to be on the trial, will they need a genetic diagnosis to be accepted onto the trial?
That's critically important. Like we need to confirm that the patient has a mutation in RPGR. Because the thing is that with retinal diseases, sometimes the phenotypes, like the back, like the imaging of the eye can look like similar. So it's really important that we have like a genetic diagnosis, like we know which gene is affected and ideally what's the mutation. Yeah, we do need confirmation before a patient can be enrolled in a clinical trial.
Well, brilliant. Okay, so we'll have to bear that in mind if you want to get involved. So again, well, thank you really again for joining us today, Cristina. It's been a very interesting presentation. It's always good to hear about where we're getting with genetics and gene therapies and things like that, because it's so important. So thanks very much for your time today.
Thank you very much.